Right. So we get to be talking about all the steps that are involved in the synthesis or formation of what matured globin from its genes. Okay. So we'll just run through it quickly. So these are the steps. The synthesis of matured globin from its genes involve what? There's transcription of DNA. So DNA is what? Moved to form mRNA. You see it there. Then there's processing of the mRNA. Say process, make it nice. Then splicing of the introns. You see it there. Then translation of the transcript into globin. You understand? So translation is usually done on ribosomes. Then it will now form globin, right? So let's talk about these steps one by one. Right. So transcription does involve creation of what complementary mRNA, which is called what primary mRNA transcript from DNA. So in man, this reaction is catalyzed by polymerase. Okay. So the complementary mRNA is a precursor of mRNA transcript that contains both introns and exons, right? So you can spend time to read the rest, right? It's usually a boring story for me. Anytime I'm reading about this globin stuff. Then processing, that means the mRNA transcript that is formed has to be processed. Okay. So following a successful transcription, okay, the mRNA transcript has to be processed, right? So at the 5' end, there's a binding of what? 7-methyl gonylate called capping, and at the 3' end, there's an addition of what? Adenylytic acid residues, as poly A, okay? And this is by the influence of poly A polymerase. Understand? So it has to be what? Okay, this is a 5' end, this is a 3' end, right? So you can just spend time to read, then incorporate the knowledge with these pictures, and you will surely understand the whole concept. Step 3 is splicing. So this involves excision of the introns from the processed transcript. You understand? So the exon intron junction are identifiable as what? Dinucleotide GT at the 5' and an AG at the 3' end. Removal of the introns gives rise to what? Matured mRNA, which is ready for translation. Okay. Then finally is translation, where the matured mRNA is now converted to globin. Alright. So here too you can actually just pause this video to spend time to look at the steps. Right. It's just to get the basic concept. All these other things is just long talk. Right. And nobody actually asks you for in-depth details like this as a student, as a medical student. Right. So this is just it for the steps that are involved in the synthesis of matured globin from its genes.